for me, envisioning success has to do with bioinformatics, okay? Because to me, every piece of information you collect either on your mouse or on your human, it's just information. And then how you use it is where the value comes in, okay? And Roger D. Filippo actually is responsible for my thinking at this moment, because believe it or not, he's one of the few clinicians that ever came to me and bugged me. Okay? He said I want a certain kind of radiological exam created, and it happens to be just a way of getting information for his patients. Okay, so if I look in the future, I look at genetic information in any sense of the word that we need to capture, right? So I want to see us build a group that can do that. Is that my specialty? No. Bioimaging informatics. I want to see that we collect all imaging informatics, 4D, 5D, whatever it is, and we collect it in a way that's useful to the biologist. So this is why I'm concentrating on creating a group of physical scientists in a team concept. Okay, so most of our collaborations are one lab, right? If you bring 3 in, it gets more complicated to make it work. If you bring 4 in, it becomes even more complicated. So my disease team grant has 6 different labs, okay, that are collaborating. And the company functions like that. And that comes to milestone-driven research. And milestone-driven research is generally different than hypothesis-driven research. And a lot of this envisioning you have to do for finalizing something through an IND to the FDA is milestone-driven, okay? So I'm going to talk a little bit about a paper that you can look at in a minute. But, so we're talking about milestone-driven. Now, how do I want to accomplish that? So Scott Fraser has been bugged lately because of a science paper. And I said to him, I'm never going to go bug Scott until I got my science as good as his. Guess what? That never happened. But what happened was, I read a science paper that said Tier 3 and Tier 2 collaborations do not hurt Tier 1 institutions. It actually increases the impact of the paper. So therefore, you should collaborate with everybody you can, because it only helps all parties. And this was not a meta study. This was not a little study. So therefore, David came to me and said, I want an high-impact paper. So I said, well, I know where to get one. Well, why don't Scott? In that process, then, I said, look, these physical science teams can help biology, can help medicine, but we have to create them in a certain way. Here's my idea, and here's my hypothesis. And this has been floated around the people at the hospital, and myself and Scott and stuff and David. But now it's time to break it out into the world. So what is the idea? Okay? You have groups in which have a hardcore emphasis, but their expertise overlaps in a way that you get all the way to the clinic with that one group of researchers. That is what our disease team grant does. Okay? Now, so what we might talk about? Well, you can imagine this in multiple dimensions. But this might be a computational scientist. This might be a biophysicist. This might be an imaging scientist. And this might be a clinician, like a radiologist, or Dr. D. Filippo bugged me to do my MR exam. Okay? So this is what I want to build. This is different. Okay? I have to create a home for the computer scientists within this world. And this is what I'm trying to do. And I will tell you that when you create this kind of group, you can bring students and physicians, MD and PhDs all together, and they can have innovation to the power of 6. Okay? And the idea then is that we bring these different basic physics, physical scientists in with disease team biologists and translational imaging scientists. And we create this group. Now you notice that David's original thing, he had this little thing called clinical incubators. Okay? That's the space that this group fits into, that clinical incubator space. Between all the groups, we have to make homes for the physical scientists, but when Tracy has a question, she wants to come over and say, should I do an SBIR? There's somebody that has SBIR experience in this group, and they can talk to her about it. Okay? And then you interact, you make them all interact together, and you can bring it outside universities. Okay? So, now, but why do I want to do this? Okay? Dewey is sitting here as our postdoc on bone development. Okay? But why did Scott and I recruit Dewey? Because David gave us the money. No, we created him before David gave us the money. But it was thank you, David, for giving us the money. But we already had him. So I work with a researcher in children's, a cancer biologist. I'm an imaging scientist. But we didn't have a developmental bias anywhere related to bone. But he was working on a drug, a bisphosphonate, okay, on the early days. Bisphosphonate drugs that are originally for osteoporosis, and I may have to hurry up here, now are prescribed prophylactically for bone metastasis. Millions of doses. De Klerk was very good. He's from Children's. He was here early. We could probably create a modification of that drug that would work better and have a huge impact on the world. So even though you didn't know it, you kind of know it, but now you've heard it again. Okay? We're going to bring 3 different expertise together, and I'm going to try and make an impact on that. Now, when we're talking about the impact of physical scientists and computational scientists, this, believe it or not, is from a sort of funded postdoc for tools and technology. That and that are the same information. This is a nuclear medicine created by a radioisotope using stellar imaging techniques, non-coded aperture pin arrays. So we basically have a radioactive source, a closer mesh, a coded mesh. You do some fancy mathematics. Ali, once your Ph.D. in mathematics, out pops our radioactive source. What do we want to do? Trap stem cells. Now this has a lot more radiation than stem cells do. But the idea is that stem cells are a lot like stars and that there'll be no background because of the very few of them. Will it work? I don't know. But we got our first real image. Now another postdoc has already done this computation, designed the system computation. Okay, here's what Roger D. Wigfall thought me about, but much to his chagrin, I took it one step further. Another postdoc, we analyze the contrast agent flow through kidneys. Okay? We take the 4D data, we cluster it together based on its 4D analysis, and we reduce the number of problems you have in the kidney to clusters of information. So instead of having 10,000 pieces of information, we have 10 or 12. Now someday I'm going to go back and sit in Roger D. Wigfall's office, and I'm going to make him tell me when he sees a cluster in a certain disease population. Okay? But it's a way of handling the information. We have Philips Medical Systems interested in it, they want to license it. Okay? This model will also do things like this. You tell me what normal is, I'll tell you where the clusters differ from normal, and you'll get a picture of the difference between normal and sick. Okay? Well, why would I do that? Where did my friend radiologist disappear to? Because I watch them. They do abnormal, normal, and normal variance. That's the 3 pieces of information I see him use to understand whether something's sick. But this is going to give Roger more information than he ever wanted, because he just wanted a couple measurements. We can take every mathematical model in literature and process it on this and give you 10 parameters for each one of those clusters. So that's probably not going to work. But we're going to eventually reduce it. Okay, now I want to talk a little bit. I told you that I have a disease team grant, and it's for neural stem cells going to tumors and treating them. But there's a nice review article that Karen and Rudy wrote. And I'm not going to go through these different methodologies, but I'll say this is ours. We take these neural stem cells, they carry an activating enzyme, and they change the drug to a more active form. This is our $20 million disease team grant. I can't go into it, but we use the trophic ability of stem cells to track the tumors, and we carry a drug with us. Now, This particular article, though, talks about how academic people can take stem cells through the IND process. Okay, I've been through the IND process. We just did 240-mile studies, and guess how many pages the report was to the FDA? 800 pages. Okay, every piece of information, you have to keep track of where your serum came from, because there might be HIV or something in it. You have to say when every bottle you used in the lab expires. Okay, this is a new world for a lot of you, but it's not that impossible, because Karen and I were not clinical. We're not pharmaceutical. So we never developed anything before that, but we're in the start. Now, sorry. Regulatory affairs at City Hall. David makes a good point. It's a whole division with a vice president in charge of it, Larry Kircher, and he's the co-PI on the grant. So we had regulatory. So just to make a long story short, the stem cell part, 10 years ago, we knew what we were going to do before the CERN grants came out. So we just happened to be in the right space. Okay, but here's the slide Scott forgot. And I totally forgot a slide. But my colleague from UCLA just happened to mention it, and I happened to have it in here. This is another dimension of imaging. So when we look at the imaging, you guys show me one slice of the console, and you try and convince me of something. Wait a minute. I come from radiology, where we like to see the whole organ, and we want you to give us a quantitative answer across the whole organ, not some one slice and tell me you know the world. Okay? So Rusty shows you all the data. Scott shows you all the data. Who's going to process that data? Well, it's going to be my computational postdoc or my mathematical postdoc, probably. And I want to make a home for those. Now, what this is it so happens that my drug that we use, Karen's drug in our clinical trial, shows up in Raman spectroscopy. So this is Scott's light sheet spectroscopy with his postdoc. I speak it at night and trying to get him to do this study, because I can then do a whole mouse. And here's a mouse. This is much deeper imaging because it's Scott's light sheet trips. And I can also potentially measure my drug concentration that we talk. So when you ask the metabolic question this morning, you notice this is yet another axis and another way to measure. So what I'm getting at is that I want to create this physical science group for you guys, not for me. Really. It's really for you guys. Because I can't possibly utilize all these people. Well, that's not true. Okay. But I think the point is that, look, if you have these people, if you have 4 or 5 of these people, that's pretty hard to keep, right? So, let's envision success. And I hope you guys will think about what it means to have this kind of group. And Scott and I will have volunteers to help.
And as you know, we're on board with this space, and completely understand that we can't get where we want to go without this. So we got 5 minutes for some queries before it's wine time. No questions? No, has anybody got anything?
Can I have a question?
Yes, you may.
Okay, thank you. That's my only question.
And you know, life is math, right? Unfortunately, I only have ordinary level mathematics. But life is math. We need to have somebody who knows how to do that.
How much dimmer do you expect the stem cells to be in your imaging?
In the nuclear medicine microscope?
Yeah.
We kind of alluded to there. Well, right now, a lot, right? So what Ali has to do is use his mathematical skills and tell me how many counts. Theoretically, how many radiation counts does it take? Less than the number of holes in my mask. Because statistically, I have to match up the pixels with that mask. So it could be as low as 100 events or 95 events. But that depends on the background. Every background is going to mess me up statistically. So theoretically, it's possible that we may make this work. And by the way, there won't be a depth problem with this. Okay, so as long as we can figure out where we are, we'll see it. But we'll also see a lot of other things, so it'll be interesting.
My other question is, should I change the locks on my lab?
Not until I get time to do that.
No, it's